Hello everyone, myself RASI. I'm going to talk about effluent treatment process and anaerobic digestions. Mm, Effluent treatment process. Effluent treatment process involves the purification of industrial wastewater by eliminating the harmful pollutants and contaminants which was present in the industrial wastewater before they discharge into the environmental or waste purpose. Why we need to do that? It is essential for avoiding the water pollutions and preserving the water resource and meeting the environmental regulatory standards. And the effluent treatment plant, it's employ the combinations of physical force and chemical and biological techniques, and which helps to remove the suspended solids and chemicals and harmful substances which was present in the industrial wastewater. In the primary and preliminary treatment, we use the physical force, but to remove the to remove the large number of particles which was present in that. Then we use the anaerobic and aerobic bacteria to break down the organic matter into the simpler molecules. Then we use the chemicals to clear out the pathogenic organisms, and it will be quality control, and it will be it will be reused in the environment or other purpose. Then we will see about anaerobic digestion. Anaerobic digestion is the process of secondary treatment process which involved in the effluent wastewater. In this process, we use the microorganisms without the presence of oxygen. The microorganisms utilizes the organic matter for their metabolic activity into their simpler convert into the simpler molecules, and it produces the biogas and transfer it into the biogas and primarily methane and carbon dioxide. The treated water with the reduced pollution level. This process is efficiently lower the BOD and CO levels and suspended solids while generating the valuable renewable energy in the form of methane and biogas. In the anaerobic bioreactor, it's carried out the sequential process. First one is hydrolysis. In this process, complex organic matter like carbohydrate, protein, and fats are break down into the simpler molecules like sugars and amino acids. Then with the help of hydrolytic bacteria, which produces the enzymes, it helps to break down that. Then the acidogenesis in the actual fermentation steps is happen here. Use the simple sugars like which was produced in the hydrolysis. It's converted into the volatile fatty acids and alcohols and hydrogens and carbon dioxide with the help of the bacteria like Escherichia and Clostridium Streptococcus. Then osteogenesis will be happen. In this process, volatile fatty acids will be further converted into the acetic acids and all. In the acetic acids and carbon dioxide these are formed. Then it will be utilized by the methanogenic bacteria, and it will be produces the biogas and the methane. Then advantages of the anaerobic digestion: energy productions. In this process, it generates the biogas and preliminary methane, which are can be used for electricity, heating, and fuel for vehicles, and helping us to reduce the energy expenses and promote the sustainable life and reduction in the waste value. And generally, greatly it reduces the quantity of sludge and organic waste and cutting down on the disposal, transportation, and treatment process. Then cost effective, we don't use the chemicals and aerations. In the anaerobic process, we use the aerations and chemicals large amount. We don't use that much amount as compared to the anaerobic digestion, so it is cost effective. Then environmental benefit, it's mitigating the greenhouse gases like capturing the methanes it would otherwise be emitted from the landfills or uncontrolled decompositions. These are the advantages of the anaerobic digestions. Then this is one of the small case study. It's about anaerobic digestions method. They examine the binary effluent, how they examine how the organic loading rate and the initial effluent concentration, impact of the COD levels, and the methane productions and yield, and the stability of the process. They compare the one stage and two stage process. From that, they analyze as the which one is best, and like this is the effluent of the red wine and white wine. In the COD level of the red wine is low as compared to the white wine, and the like other parameters are like ethanol and all. This is low in the red wine as compared to the white wine. This is the result of productions of acids during the acidogenesis. In this process, like ethanol, acetic and butyric acids and isobutyric acids in the uh, white wine effluent treatment process, they analyzes the stability, like it's like wavy in nature, it's not stable in the form, and the production is high. In normal production, is the concentration is high. Then this is two type of acid concentrations. Then the productivity of the methane productions, like it's increased, and the yield also it will be decreased. It is two-step process. First they carried out the acidogenesis in one fermenter, then the do the methanogenesis in another fermenter. Here in the red wine processes, they did in the same fermenter. First, it's stable in nature, like the acid productions and the in the stable in the form. It's high and it will be decreased. Then it will be utilized by the methanogenic bacteria, and it produces the methane, like the concentration of the acetic and propionic acid as compared to the white wine is less. This is the overall representations of the white wine effluent and red wine effluent. The white wine effluent CO level is high as compared to the red wine effluent. In the two-stage process, the first one they carried out the acidogenesis one in bioreactor, and in the methanogenesis in another bioreactor. In the red wine, they did in one bioreactor because the COD level is low. Then the organic matter removal percentage of the red wine and white wine. Red wine 93% and white wine 88%. And the methane productions are 44 and 0.44 and 0.31. We Why we need to do that? In the red wine, we have the CO level is low concentration, so that much acid will not produced in as compared to the white wine. In the white wine, the CO level is high, so acidogenesis bacteria fastly they produces the acids, so it will be inhibited the methanogenic bacteria for utilization of that byproduct, so it will be inhibited in this step. So we use the two-step process to overcome the that steps to remove that the organic matter which was present in that effluent. Next, we will see about the chemical disinfection industrial effluent treatment. In this process, we eliminated the pathogen to ensure the safe and discharge or reuse purpose. The common disinfectants like chlorine, and it's a strong oxidizer and it destroy the microbes and provide the residual protections. And ozone, it's a free oxidant. It's higher in cost, and the hydrogen peroxide is eco-friendly oxidant, less effective alone. And the AOB, like advanced oxidation process, the combinations of overall, like we use the combinations, we will resist the contaminants and all. Then the chemical disinfection process steps, the dose. Based on the chemical, we precisely add the amount of chemicals for that process to remove the pathogen. Disinfection chemicals based on the effluent quantity and pathogen load, and the contact time, allowing the sufficient time for chemicals to react and inactivate the microorganism fully. Mixing ensure the effective distributions and of the disinfectants throughout the effluent. Restitutional control, monitoring the maintenance, maintaining the optimal disinfected residues to prevent the microbial regrowth. And this is the overall view of that. The chlorination and ozonations and ultraviolet radiations. First, disinfection mechanisms or oxidations of the oxidations and chlorinations of the microbial cell membrane and enzymes. In that steps, ozonation also attack the cell wall and the internal compounds via the ozone and react to oxygen and produces the ROS, and it's directly damage the DNA and RNA, so it can't survive in that UV radiations like pathogenic microorganisms. Then effectiveness if the chlorination is most effective against the bacteria and virus, but less so for protozoan like Giardia like organisms. Then ozonation is broad range of microorganisms and including the chlorine resistant one also. Then in the UV it's against all kind of bacteria and viruses also, and but only it can affect you with the clear water. If it is turbidity, the light it cannot penetrate into that. Then the disinfections by the products, it produces the like trihalomethanes, which is harmful to us as carcinogenics. Then in the ozonation, it's may form the bromide. It is harmful to us. There is no chemical byproduct is formed in the UV radiation, and the restitutional productions provides, so chlorination provides the restitutional productions, but ozonation and UV can't do that. Then the required time, it's moderate level. It's a very short and rapid disinfections. Then UV radiations, we uh, instantly we will degrade the all organisms. This is the overview of the process. Like if we alone we use the chlorine, we need to increase as the chlorine level, then only it's completely remove that pathogenic bacteria. Like that, it will be tailoring phenomenon. It will be present in after we disinfected that, it will be present in that water. It will be harmful to us, but if we enhance that process by using the combinations of UV and chlorine and all, we can effectively remove that pathogen load, as well as the as well as the as well as the avoiding the tailing phenomenon. Like this is the graph of that. This is the inactivation rate, and this is the green color represent the color of that effluent water. Then is the turbidity rate, it will be reduced from that. Like we use the sequential like that, it drastically reduces the level and inactive rate of the pathogens. It's the importance and benefits of the chemical disinfections to ensure the complete microbial safety, to treat the effluent, to comply with the environmental and discharge regulations. It's integrated with the other treatment methods. It enhances the efficiency, then protective the aquatic ecosystem and by, public health by removing the disease-causing pathogens, and allow the safe use of the treated water in an industrial process and irrigations or cooling system. These are the references. Thank you.